Assalamu alaikum, dear students. I am Muhammad Rauf, lecturer, Department of Zoology Cast. Welcome to the course Principle in Animal Life 2. This is lecture number 30. The topic is Applications of Recombinant DNA Technology. Discussing agenda for the topic will be introduction, medical applications, human gene therapy and gene editing, pharmaceutical products, protein production in cell cultures, agricultural applications, protein production by farm animals, forensic evidence and genetic profiles, environmental cleanup. Lecture outcome. After watching and listening this lecture, students will have an idea about the contribution of recombinant DNA technology in improving the quality of human life. Introduction. The practical applications of DNA-based biotechnology affects our lives in many ways. They have contributions in various fields, just like we discussed in the uh, lecture agenda. They have contributions in medical sciences, contribution in forensic sciences, contribution in agricultural sciences. So they have a lot of contributions in the world around us. DNA technology is in the news almost every day. Medical applications, identification of human genes whose mutations play a role in genetic diseases. These discoveries may lead to ways of diagnosing, treating, and even preventing such conditions. Means if we identify a particular gene and then know about its function, the mutations which can affect it and which can in turn affect their product and the inheritance of such genes which are more vulnerable to mutations. So this can help us in diagnostics and treatment and even prevention of such disorders. Recombinant DNA technology is also contributing to our understanding of non-genetic diseases, from arthritis to HIV/AIDS, since a person's genes influence susceptibility to these diseases. As you know that genes are under the effect of environment. Sometime a gene is only expressed under a particular set of conditions, and there are certain genes which are vulnerable to deterioration, which are which are vulnerable to mutagens like proto-oncogenes. A new chapter in the diagnosis of infectious diseases have been opened by DNA technology, in particular the use of polymerase chain reaction and labeled nucleic acid probes to track down pathogens. All of you must have learned the word PCR in the last 3 or 4 months, which is used for the detection of various disease-causing agents, and the tests of the coronavirus are performed using PCR. For example, because the sequence of the RNA genome of HIV is known, RT-PCR can be used to amplify and thus detect HIV RNA in blood or tissue samples. With the help of RT-PCR, we can detect the presence of HIV in a person. Furthermore, diseases of all sorts involve changes in gene expression within the affected cells and often within the patient's immune system. By using RNA sequence analysis and DNA microarray assays or other techniques to compare gene expression in healthy and diseased tissues, researchers are finding genes that are turned on or off in particular diseases. We identify a particular cell and then take out all the RNAs from that particular cell, and then we identify those RNAs and we compare the expression pattern of that particular cell with other cells. These genes and their products are potential targets for prevention or therapy. So, in order to find a cure for a disease, for a problem, first you need to identify the various ways in which the disease affects the individual. You should be aware of the pathogenesis, and only then you can devise strategies for prevention or cure. Among the genes for human diseases that have been identified are those for sickle cell disease, hemophilia. Sickle cell disease is a condition in which the shape of the RBC becomes sickle-celled. Hemophilia is a condition where a blood clotting factor is missing. Cystic fibrosis is a congenital genetic disorder where a sticky sort of substance prevents the alveoli in the lungs from inflating. And Huntington disease is a neurological disorder, and it's a muscular disease. So the genes for all those diseases have been identified. Individuals with such disease can often be identified before the onset of symptoms, even before birth. Now, the genes of those diseases have already been identified. Now, we can find out which person will be vulnerable to acquire those diseases in future at the time of birth or even before birth. We can find out whether a person born will have any genetic disease or not. You know about prenatal testing, means testing before birth, which is amniocentesis, chorionic villi sampling, pre-implantation genetic diagnosis. So all those are prenatal tests which let us know whether the person to be born will be suffering from any kind of genetic disorder or not. And even if we miss to carry out analysis or tests during that gestation period, after birth, shortly after birth, Through postnatal testing, we can also identify whether a person is carrying such a gene which makes him or her more vulnerable to acquire a particular diseases later in life or not. So PCR can also be used to identify symptomless carriers of potentially harmful recessive alleles. You already know about the dominant and recessive alleles. So this PCR polymerase chain reaction is used to identify individuals who are themselves healthy, but they carry a defective copy and could transfer it to the next generation. As you learned earlier, genome-wide sequencing have pinpointed SNPs that are linked to disease-associated alleles. So these SNPs, these are specific sequences in the genome which let us know about their association with diseases. Knowing the expression levels of particular genes can help physicians determine the likelihood that the cancer will recur, thus helping them design an appropriate treatment. So now the researchers are talking about the person, individual specific treatment. So they can study their genes. We all are different from each other. Our genes are different from each other. We respond to the environment in different ways. So by studying genes of each individual, we can devise person-specific strategies for treatment. Human gene therapy and genome editing. Gene therapy, the introduction of genes into an afflicted individual for the therapeutic purpose. A person is suffering from a genetic disorder. Suppose the person has diabetes or other genetic disorder, and gene therapy is a technique where we can introduce a healthy gene into an organism to replace the defective gene, to minimize the effect of the defective gene. This procedure is called gene therapy. The aim of this approach is to insert a normal allele of the defective gene into the somatic cells of the tissue affected by the disorder. Now, a more direct approach that avoids the complication of using a viral vector in gene therapy is made possible by gene editing. Now, the therapeutic gene or the gene of interest is usually loaded upon a vector, a virus, and then that virus is introduced into the human body so that this virus along with the therapeutic gene can uh, introduce that gene into the genome of the individual, and that gene becomes a permanent part of the genome. But quite often those viruses causes insertional mutagenesis. Quite often those viruses results in a severe immune response. So the researchers are thinking alternative ways in order to introduce the gene of interest into an individual. And one of the latest technologies in this regard is CRISPR-Cas9. CRISPR-Cas9 is a technology which was initially studied in bacteria. The bacteria, it is part of the adaptive immunity of bacteria. In this technology, A gene can be introduced into a cell and into the DNA with 100% accuracy in vivo inside a living cell. Now, these are the various pharmaceutical products that have been formed using the recombinant DNA technology. How they are formed? They are formed by first of all obtaining the gene which synthesizes those proteins and then introducing that gene into other organisms where it can be expressed. For example, erythropoietin is a protein which is responsible for the formation of RBCs. Now, if this protein is lacking in an individual, the person suffers from anemia. So what the researchers have done is they have isolated that gene which codes for that protein, and they have inserted that gene of interest into E. coli and cultured mammalian cells. Now E. coli and cultured mammalian cells are producing this protein which can be purified and which can be used as a treatment against this condition. Similar is the case with other genes. They, the middle column is showing the effects of the absence or abnormal form of those proteins on the body, and on the right side the organisms where those proteins are being cultured is given. Protein production in culture cells. Pharmaceutical products that are proteins are commonly synthesized on a large scale using cell cultures. You learned earlier in the chapter about DNA cloning and gene expression systems for producing large quantities of a chosen protein that is present naturally in only minute amounts. The host cells used in such expression systems can be engineered to secrete a protein that it is made, thereby simplifying the task of purifying it by traditional biochemical methods. So before the production of insulin through recombinant DNA technology, there were still people who were suffering from diabetes. They were in need of insulin. So, before the production of insulin through rDNA technology, the doctors usually obtained insulin from the blood of cadavers. Cadavers means people who had passed away or from other animals, and the consequences was that the insulin or the protein of other animals often provoked immune response in humans, and there were more people in need of this protein, and the production was very low, and furthermore the purification process was extremely difficult. So now, be courtesy of recombinant DNA technology, this protein, the and other types of proteins which are important from therapeutic point of view can be produced in large quantity and purified easily, employing the natural systems. Among the first pharmaceutical products manufactured in this way was human insulin. It was produced in 1980s and it was called Humulin. Humulin means human insulin and human growth hormone. Some 2 million people with diabetes in the US depend on insulin treatment to control their disease. Human growth hormone has been a boon to children born with a form of dwarfism caused by inadequate amount of human growth hormone. This hormone is responsible for increasing the your height and for proper shaping of your body, and the absence of this hormone results in dwarfism. So even if a person is lacking the functional form of this hormone, then courtesy of recombinant DNA technology, the person can buy this hormone from a medical store. These are the fishes which are bioindicators, means these fishes are genetically engineered. They carry a specific gene which is turned on only when exposed to heavy metals or other impurities in water. So they are a kind of bioindicators where you doesn't need to take water and test it for the presence of a harmful substance. So the fishes, the organisms living in water will let you know about the quality of water. So this was world's first GM, means genetically modified. The scientists and the scientists faced a lot of criticism for developing such a fish. Agricultural applications. Scientists are working to learn more about the genomes of agriculturally important plants and animals. For a number of years, they have been using DNA technology in an effort to improve agricultural productivity, means to develop such crops which are resistant to pests, which are resistant to harsh environmental conditions. DNA technology enables scientists to produce transgenic animals which speed up the selective breeding process. Selective breeding is a process in which two organisms of a species having favorable traits are crossed with an intention that the offspring will impart the beneficial characteristics from both parents. It is a process which has been taking place for thousands of years, and it is a process which requires a lot of patience. There are chances of failure. The individual's lifespans are very long at times, and it is very difficult and time consuming to know about the success of a cross. So it is a hit or miss method. You cross best with the best and hope for the best. Sometimes the breeding experiment pays off, other times it fails completely. So in order to avoid such problems, now the scientists can directly incorporate, insert the gene of interest into an organism, and then in a very short span of time they can analyze the success or the failure of their experiments. The goal of creating a transgenic animal are often the same as the goal of traditional breeding. For instance, to make a sheep with better quality wool, a bull with leaner meat, or a cow that will mature in a shorter time. So scientists might, for example, identify and clone a gene that causes development of large muscles in one breed of cattle and transfer it to other cattle or even to sheep. So it means the scientists can identify an ideal gene in one species and then they can transfer that particular gene to another species so that the other species can also acquire the same characteristics. Agriculture scientists have already endowed a number of crop plants with genes for desirable traits such as delayed ripening, resistance to spoilage, disease, and drought. So some transgenic plants have been developed which has such type of characteristics against the environmental circumstances. For many plant species, a single cell grown in culture can give rise to an adult plant. They are talking about tissue culture. The genetic manipulation can be performed on an ordinary somatic cell, and the cell then used to generate a plant with new traits. So genetic engineering is rapidly replacing traditional plant breeding programs, especially for useful traits such as herbicide or pest resistance determined by one or a few genes. Crops engineered with bacterial gene making the plant resistant to an herbicide can grow while weeds are destroyed. So means the certain genes have been identified in bacteria and those genes are resistant to herbicides and those genes have been introduced into plants which are beneficial, which need not to be destroyed with herbicides. So because of that particular gene, such plants, such beneficial plants are protected from the effects of herbicides and pesticides which eliminate weeds and other unwanted plants. In India, the insertion of a salinity-resistant gene from a coastal mangrove plant into the genome of several varieties of rice has resulted in rice plants that can grow in water 3 times as salty as seawater. So these experiments were performed Coastal mangroves are the areas which are near a sea. Usually those areas have water standing throughout the year, and sometimes certain plants also grow in those water-laden areas which are called mangrove plants. So the researchers studied the genome of the mangrove plants, that how such plants can thrive in that salt-rich water, and then they identified a gene which imparts them that ability. They isolated that gene, and then they introduced that gene into rice. And now the rice, because of that introduced gene, is exhibiting the characteristics of that gene, and that gene insertion has enabled the rice plant to thrive in water that is 3 times more salty than seawater. So this is how the transgenic plants or the gene insertion techniques are imparting useful characteristics to organisms within the blink of an eye, instead of waiting for several generations for breeding results. Protein produced by farm animals. In some cases, instead of using cell system or to produce large quantity of protein products, pharmaceutical scientists can use whole animals. Now, instead of using bacteria or other simple prokaryotes or eukaryotes to produce proteins, the scientists are using animals to produce large quantities of those proteins. And they are a kind of living factories where they produce proteins. So such animals which are introduced with a gene of interest, and then those animals produces that protein in their milk or meat or eggs or other products. So such animals are called farm animals. And the gene of interest is usually inserted in those animals when they are just a single cell. Or the gene of interest can directly be injected into their fertilized egg. So this transgene or the gene of interest or the introduced gene becomes part of their genome, and if the insertion is successful, then this gene expresses itself into the organism. A transgene for a human blood protein such as antithrombin, which prevents blood clots, can be inserted into genome of a goat in such a way that the transgene product is secreted in the animal's milk. Now, how unbelievable is that? Human blood protein antithrombin, the gene for that protein has been isolated and it has been inserted into goats, cattle's, and now the cattles are producing milk having this protein. So such type of animals are known as farm animals. The protein is then purified from the milk, which is easier than purification from a cell culture. Such proteins must be tested to ensure that they will not cause allergic reactions or other adverse effects in patients who receive them. forensic evidence and genetic profiles. In violent crimes, body fluid or small pieces of tissues may be left at the scene or on the clothes or other possessions of the victim or assailant. If enough blood, semen, or tissue is available, forensic laboratories can determine the blood type or tissue type by using antibodies to detect specific cell surface proteins. Now here we are discussing the contribution of recombinant DNA technology to law, to justice, where the individuals killed in an incident can be identified. Suppose an aeroplane was crashed, then you can take samples from the site, and those samples can be used to identify the individuals who were part of that tragedy. And suppose several children were born in a hospital and they somehow mixed together, and now a particular family is unable to identify their offspring, so they can carry genetic tests. And in other instances, suppose a person was murdered, and the killer left some of his marks in the form of a hair or a blood drop, etc. Now, this evidence can be used to trace the culprit. Similarly, the victims of murders and the individuals who carry activities like rape, etc., can be apprehended through those techniques. The term DNA fingerprinting, just like we have a fingerprint, similarly, our DNA sequences are unique to us. They are different from other individuals, except monozygotic twins. So, this unique nucleotide sequence in DNA can be used to recognize an individual. Environmental cleanup. Now, the diverse abilities of certain microorganisms to transform chemicals are being exploited for environmental cleanup. At times, there are oil spills in the seas which are very difficult to clean, and they poses lot of threat to the aquatic life, and similarly there are certain agents which are harmful to the environment, for example plastic. So what is the contribution of genetic techniques and recombinant DNA technology in this respect? Such microorganisms make them unsuitable for direct use. Scientists can now transfer the genes for their valuable metabolic capabilities into other organisms, which can be used to treat environmental problems. For example, many bacteria can extract heavy metals such as copper, lead, and nickel from their environment and incorporate the metals into compounds such as copper sulfate or lead sulfate, which are readily recoverable, which can be recovered. So these bacteria extract those heavy metals from their environment, and they can be used for the environmental cleanup. Genetically engineered microorganisms may become important in both mining and cleaning up highly toxic mining wastes. Biotechnologists are also trying to engineer microorganisms that can degrade fluorinated hydrocarbons and other harmful compounds. These microorganisms could be used in wastewater treatment plants or by manufacturers before the compounds are ever released in the environment. This was the end of the lecture. Thank you very much. Allah Hafiz.